Hello everyone, good evening, and welcome to panel discussion tonight on art and science in dialogue. And we have 5 collaboration partners here on stage tonight. Very welcome to you as well. My name is Sarah Donder. I'm curator here at ZKM since 2020, and I've been working on several projects on the convergence of art, science and technology. And recently also co-curated the Renaissance 3.0 exhibition, which is currently on display. ZKM is focusing on the collaborations on art and science, or between artists and scientists, for several years now, or centuries, and especially focused this year as well with the Renaissance 3.0 exhibition, where we are inviting or opening a base camp for new alliances between art and science in the 21st century, and also the Driving the Human project, which is also opening tomorrow in the 3rd part. We are presenting 7 tangible prototypes for eco-social renewal. And I think like today also this is end of a longer day already for the panelists and also some of the audience, because we co-organized together with the Sharing Stiftung workshop or participatory forum for 2 days on exactly art and science collaborations, and we want to try to establish a new research agenda on that. So thank you that you're still with us today, because it was, you see, we are still in the working space. In that moment, I also want to thank Sharing Stiftung for the really fantastic collaboration. And today also we have the team and around Katja Nahe here. So welcome to you as well. In this framework, I'm really very delighted to welcome these 5 participants here on stage that have collaborated on and working together on the project Fermenting Futures. For like the last 3 years or almost 4 years, right, before the pandemics. And this project is also on display currently here in the exhibition Renaissance 3.0. The project started right before the pandemic, which also had some obstacles, which we will address today, I guess. And it focuses on different yeast research strings. So I think this is something also we will dive in deeper. And the basic idea is also for the project is the importance of yeast for the society, for example, development of settlements, and also for the future. So how, for example, yeast could help with carbon bindage or also biodegradable plastics. And just give a brief introduction to the panelists, because I think it's better also for you to explain what you've been working on. Next to me is Diethard Mattanovich. He's a professor for microbial biotechnology at the Institute of Microbiology and Microbial Biotechnology at the University of Natural Resources and Life Science, short BOKU, in Vienna. And he's been investigating the application of yeasts for the conversion of carbon dioxide and other renewable feedstocks into chemicals of our daily use, such as biopolymers. So, very welcome to you. Also, we have Dumitriu, British artist with us, who works with bio-art, sculptures, installations, and digital media to explore our relationships to infectious disease, for example, synthetic biology or robotics. And your works have been already exhibited internationally at various venues, including ZKM, but also, for example, Victoria and Albert Museum, Oxford Science Museum, or MoCA Taipei. And also next to you is Alex May. You've been working with him for several years now on different projects. Alex is also an artist from Britain who uses digital media to explore physical and emotional human boundaries on a personal and social level. And you've been questioning like the collective experience of time in different ways, and also with various media. And next to you is Sonja Schachinger, who I welcome to you as well. She's an Austrian independent producer, curator, and art science mediator. And you've been for many years working in international exhibitions and also working on the convergence of art, science, and technology, and also with the focus on digital and multimedia art. I'll just mention it because maybe we will come across later on. You've been directing the mediation format We Guide You at the Ars Electronica Festival for 4 times already. And maybe we can discuss later also the role as mediator. Yeah. And next to you also welcome to Ingeborg Reichle. She's an art historian on contemporary art and cultural theorist and currently senior fellow at the Institute for Advanced Sustainability Studies in Potsdam, Germany, and you've been writing and publishing about art and science collaborations extensively already in the last few years, and you've been also in the recent years a professor in the Department of Media Theory at the University of Applied Arts in Vienna, Austria, and you are founding chair of the Department of Cross-Disciplinary Strategies. So, well, welcome to you. Before we start into the broader discussion about art and science collaborations or the dialogue between art and science, I would like to give a bit of an insight into your recent collaboration, and I think it's the best way if you answer that. So I would like to know about the frictions, the questions, and the takeaways of your collaborations that you came across. So I'm just starting very quickly. So maybe in one word or sentence, what does yeast mean to you? So everyone could start. I think they're on, yeah.
Well, that's a difficult question for me because yeast means kind of nearly everything of my research life or my more recent research life. Yeast practically has shaped human society to quite some extent, and we might dive into that string at one point. Yeast is making our bread, it's making our beer and wine that we might enjoy after this event. What else? Yeast biotechnology will have a bright future. I dare say there is a lot of applications that are now in the fermenting, and I'm using this kind of pun, that might really have a huge impact on the way we are producing goods, possibilities to wean us from let's say, the oil and gas dependency. And I think I have said too much already, sorry.
I guess for me it's something that's different to bacteria, because a lot of my background is working with bacteria. So when I first started working with Diethard, it was a kind of learning curve to understand this thing which actually has sex and things like that. So it was something that I had to get my head around, probably.
I would probably say and it's like architectural, the colonies that we scanned and the structures of them were just, kind of amazing to sort of explore through the artwork and just, yeah, it's just really, when encouraged in that, in a Petri dish, they just formed the most amazing sort of self-expression.
Yeah, okay. For me, it started with Anna, that yeast becomes a deeper meaning for me. And in general, I learned that it's a fungi. So I'm every day learning with this group.
Yeah, I was quite intrigued when we started to talk about yeast and how human civilization, or how the way in our project we talked about the co-evolution between humans and yeast, And for me, it was interesting to see that there were quite a lot of artists in the 60s and 70s working with yeast and sugar and other materials, so-called living materials. And then we had, transgenic yeast that is almost capable to save all the global challenges, especially in your lab at Diethard. So I was very intrigued to get more familiar with those issues through this project.
Yeast, as you already mentioned, takes a big role in your life and research. So, as I understood, right, you started with the idea of collaborating with artists. So it was your first collaboration on that sense. How did you come across this idea, and also because it came from Boku, the university? What were like the first obstacles that you had with like your idea to collaborate with artists?
Yeah, so probably the whole project started in kind of an unconventional way. So as you said, it emerged in my head essentially. So as a genuine interest in arts and then the interest in how, let's say, such a collaboration, what that would mean, how that would kind of change our work in the lab or impact on it, and I was pretty not aware or not sure of what exactly would happen, and honestly speaking, I didn't care so much. I wanted to see what's gonna happen, and one kind of initiation point was then a chance of having such a collaboration being embedded somehow in a large conference that we were planning, the International Yeast Conference. So that was kind of the starting point, and at some point I came across Anna's artwork on Twitter. So that may sound funny, but it's very serious. I saw that, and I really I was intrigued, and I was diving into details, and at one point contacted Anna and asked her if she would be interested to do something with yeast, and the reply was like, yeast what? Let's talk. Yeah. And I hand over to you now, and I get back to the second part of your question then.
Sure. Because Anna, well, how was it, or Alex, how was it for you then when Diethard reached out to you?
So what happened then was I said, well, I've got this show on in Oxford, because I had a show at the History of Science Museum, solo exhibition, Bioart and Bacteria, not yeast, there. But I did have some yeast in the show, because I forgot to say earlier, I'd done a little bit with yeast in the Laboratory for Synthetic Evolution, which was a residency that Jens Hauser, who's here now, was co-curator of with David Vermillion at the University of California, Irvine. And they were working with speeding up the mutation rate of yeast in that lab, so you could use it for things. If you could speed up the mutation, it would do things faster. And I really like that idea of like speeding up mutation, because it seems so wrong that it wouldn't be something you want to do. So I actually had that on in the show. So Diethard was able to come over, because you kind of came over to see the show and saw that work, and then we talked. Simple as that, basically.
Nice. And Alex, for you, how was your first thought when like yeast project came up? Also from like your kind of approach to also scientific research.
Well, I mean, same experience. Meeting Diethard at the exhibition, and then just we discussed it, and we sort of. Just embarked on the journey and started learning about it and going to the lab, and it was there's nothing quite like going to the place and seeing where the work takes place and with what tools and what processes, and just seeing the science that takes place on a daily basis, and having the conversations with the scientists, and it's just began the process of developing the work.
And then Sonja came into play, right?
Yes, maybe make a small introduction for that. I was invited by Anna to join this team. Maybe Diethard can give a small comment how he felt without me.
Yeah, you asked for the difficulties, so the first steps were super easy, I have to say, and I think we immediately realized that like we have a lot of common interest and kind of really this thought of being intrigued by diving into that new world from your side, the yeast world from me, the like art science collaboration. That was very clear, and I think we never had a doubt that we are gonna do it, and kind of how we would approach it. Next step, raising funding. It's more a technical point, I have to say, but very important, and we can talk later about that. But then the next step was like where to show it. So we had this conference, which in the end did not happen due to COVID, but or it was an online event. But that was one point. We could show it there, but then looking for an exhibition space, I was probably quite naive, like contacting different museums and using some contacts also to talk to like Natural History Museum, etc. And no one took me serious at all. So I could see in the faces that no one simply took me serious, and I realized, we definitely need someone professional on that. And then Anna suggested to invite Sonja, and that's how you came into
how the story for me began. It was clear that I don't need to mediate between science and artist because they are on eye level. So we have with Anna brilliant expertise, and she knows what she's doing. They know that they want to do something, and but for making an exhibition, to make the mediation for everyone getting involved, to have all these layers, the team was big from the science side, and also it became bigger and bigger from the art side. I had a co-curator, Wolfgang, who is today not here, who is an expert in developing exhibitions, so exhibition design and all these skills you need for that. And it was also really important to get Ingeborg. And I didn't know her at that time. So I was kind of like, okay, Frau Professor Reichle, can we have a coffee together? And it was really like that. So it was not just a joke. It really happened that we had a coffee.
Yeah, and I think that what was important that at that day or that time we had in the art school in Vienna, we had a bio lab for the students, a bio art and bio design lab, and there were a number of DIY bio artists around that were also interested in yeast, working in yeast. And so for the students, but also for the staff in my department, Anna was a great inspiration, coming in 2019 into my class, giving lectures, and most students were quite aware of her art, also because of her fantastic website. And so this, I think, really turned out to be a very interesting way to bring the students in contact with an artist, and also then with Alex, that is in the field for so long, always looking at new challenges, going into other materials, now yeast, before bacteria and other topics. So I would say for this, it's a small community, so it's not like big, like a huge class like architecture or painting or so. The bioart class was small, but as I said, it was an inspiration, and also to follow in conferences like TTT how the scientists and the artists, both as a team, how they collaborated and how things turned out quite nicely.
Yeah, and I think also it was an extensive exchange with the students on this topic, and then also later on with a student at the Boku University. So it was already like a follow-up, I would say, right, in that sense. And I'm curious, because diving into your project, I've also seen that you combined two different strengths of research at the Boku. So the one on the genetically modification technique of yeast to like capture carbon, but then also on the other hand to produce like biodegradable PLA, so polyphenol. So how did you engage? How was your exchange? I mean, it was an obstacle, of course, with the pandemic that you couldn't like have site visits all the time. But how did you exchange on that level? So was this like a really debate just on yeast, or then really already the two strings, or did you bring that together?
If I may start, what I did before we had your first visit was to invite all people in my lab to express their interest in that project. So no one was forced to, like PhD students may have busy times and difficult experiments and not having time for that. So only those who really had the time and the interest then got kind of slots to talk to you. So you had a hard job to have all these science talks, and we gave you quite some food for thought, I would say. So all the different projects were presented and discussed, and then we were talking about, you were digesting and bringing up these topics, and that specific PLA story. There are two different lines of projects in our lab. Very briefly, one is to make polylactic acid, bio-based plastic from sugar or from starch. This is kind of what we would call the first generation of a bio-based plastic, and this is very mature. So it's I would say close to industrial realization. And the other is this CO2 capturing yeast, where we brought the same metabolic pathway in, so we can make many amounts of lactic acid from CO2. And meanwhile, we have the proof that it's really based on CO2 alone. And discussing with the people, we knew, of course, of the two projects, but we didn't really, think them together. And then Anna suggested to bring that all together into one story, where we finally have the PLA, the yeast-made PLA, with some of the CO2-made PLA, and this as a physical piece as part of the artwork. Yeah. Over to you.
So, it sounded fascinating to me, the idea of capturing the carbon in the yeast, and then this story of the plastic, because, we've got this problem with plastic, so we shouldn't have more plastic. But then if you can capture the carbon from the plastic biodegrading, and it's actually biodegradable, which it is in 6 weeks, if you compost it in the right way, it will fully biodegrade. So it's like, maybe we could, it's a kind of, it's on the verge of speculative, because it's still quite technically complex, but you have it working in some form, don't you? And not as successful as the one from the sugar, but it works in some form. And it's just like this idea that if you captured all the carbon and you made plastic with it, and then you could capture the carbon from the plastic biodegrading, it's like you've got this like dream solution, and we could literally drown in plastic as much as we want. So we wanted to kind of throw this idea out there as like, you know, it's because the way it's always presented is we must change this, but there are potential biotechnological solutions as well that might achieve something or point to it. So we wanted to point to the possibility, I guess, of this, which sounds almost quite wrong. But I like that. That's the thing with art, is that it can suggest things that seem like counterintuitive, and then you have to think about where that might lead, what that might mean, and whether that might mean a potential future. So that's why it's called Fermenting Futures as well.
It is also interesting how you went public with the project, because it's very complex, also the two scientific researchers. So my question is, you said you worked together also with a colleague about the presentation, and also how to scenography-wise you can realize such a project and contextualize.
Yeah, this was really an essential part. We had, on the one hand, the residency funded, but nothing more. So it was something I developed with Ingeborg, to have this discursive exchange from the art side also to the museums, because it was really challenging to bring Anna's works to a real art museum. Bringing it to Künstlerhaus Vienna, it was really that. I think we had several meetings there explaining that nothing gonna happen to all the other artworks. You need to think at the same time it was Weiwei, so and it's like, can there anything go wrong? And we said, no, there is nothing gonna happen. I mean, we had soil. Yeah, we had soil. And one thing was also, I said in the workshops today, we needed to shift a little bit in plannings. So I'm really happy that one group today wrote, the knowledge group, what to do when you make art science collaborations. You make an exhibition, you make texts, you make a book. Yes, true, you make a book. And we also wanted to make a book because we really worked hard. We had many, many team meetings. We had so many people involved. And everyone wanted to share what we learned, the exchange, our change, what Anna changed with us. And then this didn't happen. And this was also part then how to make a transmediality of a book on these experiences. And we did it in the exhibition. So therefore we have the part, like you in small, that we have this science part and also these artworks. And then we had a frame program where we tried really to involve everyone. Maybe you can add things, what came out of your lab.
Yeah, I think what was particular, or what was for me particularly interesting in this project was that there was a nucleus, that we had two artists and a scientist that were like, this was the way where all the other electrons were like gravitating around, but there were a lot of like offsprings, project like Biotechnologie ist kein Kinderspiel, biotechnology is not a children's play, where Diethard and Sonja and I went to, I don't know how many institutions, but especially we went together to one just to ask for more funding and things like that. But I think we were spreading the word about art and science collaboration at Boku and with colleagues from UK within the institutions in Vienna to raise some money. And what happened was that there was then extra funding for hands-on workshops that Günther Seyfried did, a bioartist from Angewandte, And so, you know, from this core idea of having an exhibition, also showing the scientific research from the lab, and also giving lay public the opportunity to do something hands-on. And we also had a video on how transgenic geese in the lab of Diethard worked, and. So this was the starting point. I think Fermenting Future is much more than we see in the exhibition. In the end, it was a kind of community that was, going around this topic.
And I want to add something from these times of fundraising, talking to institutions. I also talked to my colleague Reichle from Wiesbaden, and she took the topic of fermenting futures and showed it in combination, I think, with the CHIC project. I'm not.
Also with Make, Do, and Mend.
Yeah, but in general, we tried to bring it to a brighter cooperation. We had different exhibitions, but she also did a discursive format, like we did it, with panel discussions. We had radio shows. We had the TV. I mean, it was, I think, more like when we were contacting all the institutions that they said, like, oh, Corona, and you had already so much money, like we want to give also to other projects. I honestly tell the truth. And we said, okay, but the public came by themselves. So we could really make like in-kind cooperations, which brought the project also to the public. Yeah, it was really great.
I think it's also important to mention that Heike Sutter and her colleague also organised a course with Goethe University based around the projects, and the students there actually co-curated the show at the Kunsthaus Wiesbaden. So we had this big show of it there, and Diethard had a painting in the show as well, didn't you? So.
Oh yeah. I'm really humbled by that.
Yeah, absolutely.
Yeah. If like artists, scientists, is there like this distinction or also the interest, right, on both sides? But also, regarding the public appearance, you said also you could present a project on the yeast conference. It was online, but how was the feedback from the scientist side?
It was online, and say from participation side, it was a huge success. And actually, this project that was kind of a public session. So we opened it to essentially everyone interested. The people had to register. They brought in a lot more people also. And feedback was great on the science side, and also as far as you get that from when you don't meet people from public participants. I don't know if you got any more feedback on that, but what is intriguing me is that a lot of my colleagues in the yeast community, which is not huge either, still talk about that, and so it really changed kind of also an approach or attitude or something, or at least people talk about something that they might have had in their minds and an interest. And I see a change there really in our rather small community.
It's like really perfect outcome, let's say, right? But it sounds very perfect. What were also the frictions maybe, or the misunderstandings that appeared in your collaboration? And not if there, because it appears really great outcome. For example, also Anna and Alex, you started like years ago to work in labs. So also from your experience, what were like really the problems and frictions? Because I think it's also interesting for our audience to see, okay, how are the beginnings of those collaborations, and how could we approach them?
Similar to how we started this one was this intense period in the lab, just like meeting all the scientists and trying to get head around the research. I've had experiences like that before. I mentioned the wetware project. That was very much like that, embedded in the lab, really intensive kind of like, I get out of the lab and then Alex says like, how was that? And I was like, I can't speak. And then I just sort of stare into space for a few hours. So I guess it's a bit like that every time when you're really trying to come up to speed. And then in other ways with like microbiology in terms of bacteria and infectious diseases, because I've worked with that for a very long time, I'm coming in at a kind of different way when I'm talking about stuff like that, because I can kind of come in at quite an experienced level. So it's sort of different in various ways. So, I mean, frictions. I mean, I don't really have that much in that sort of direction of the frictions. It's more in the like deadlines and getting it done and getting it out there to the public in the way that we want, you know. And it's really nice when it, like, there was a comment earlier about footfall being something that the funders want. It's something I want as well. I think it's something that you want, because you want as many people to see it, and people that aren't from that background as well. So it's great showing art in a yeast conference, because they don't get art all the time sort of presented to them. And what we did was we filmed that. We hired the Regency Townhouse in Brighton, which is a historic house, and we filmed it in their kitchen, which is the absolute stunning space, and filmed the whole video of the work and made it specially for the conference to present it properly, so they had something of very high quality to show it. So that was all quite stressful, because it was still during sort of just coming out of COVID, so there was that kind of thing. But I don't have that massive issues with that sort of stuff and those kind of frictions. It's interesting. I mean, I guess with this one, there was this whole other world of the project going on with Sonja that we didn't actually get to directly hear about that much. We would have occasional meetings with Sonja, but then there were bits that I didn't know about.
I said it in our talk before. I really learned much on contracts in an art museum and bringing new media art to an art museum. They have different rules. Yeah, this is really something I learned. Also, this was a challenge for me. Yeah.
And for me it was interesting to see, because one of the key questions for me is, when I look at artists going into laboratories, why do we need, within the discourse or within the narrative, the so-called bioartists, who is capable of doing all those hands-on protocols and having the ability to work in a decent scientific lab? Why do we need that within the narrative? And I understood after a while, because I thought maybe because there was a pandemic, it wasn't, the residency wasn't as we planned it, or as it was planned. But on the other hand, I think it was important, because I had other conferences before on art science and technology, art science and philosophy and things like that, and we had all these topics and debates and so on. But they were all focused on language and text and hermeneutics and continental philosophy and so on. And with the artists getting involved, we had a different, I would say, a different language. There was a visual language all of a sudden, which brought, and this is what Diethard said, and maybe you can elaborate on that, this brought a very different, I would say, perspective on the whole project. Because of course we were reading texts on, bioart and so on, but if you have the artist and the perspective of the artist in the lab with the postdocs and the students and so on, the actual object or the process of doing the object is very different from text and from discourse. And this was, to me, a very interesting, I wouldn't say media theory concept, but yeah, and to be so close to the production of the group and of the artwork, I think was very, very interesting for me.
It wasn't just a visual thing. I mean, I did a project at the Francis Crick Institute in London, which is this enormous centre of genomic research in the UK. And I was doing these lab visits where I had like an hour in one lab, and then that was it. And I just had to go in there and absorb and record as much as I could, because and that's everything from the aesthetics of the lab to the smell to the just the feel of the lab coat. It just became this very experiential sort of process. So when we get the opportunity to sort of spend a little bit longer and but it's still the same thing. We're still looking at like the materials, like the what was it, the palm, the palm tops, the dried palm tops that you were using, and that we could sort of collect for materials. It's like that point that you spend in the lab, that is a sort of very special time when you have the conversations, you're gaining knowledge, but you're also gaining a sort of physical experience of being there, and that informs the art, and it informs the whether consciously or subconsciously, it becomes part of the process. So I think that was the thing that with the COVID is that we were there just before the lockdown in Vienna. We did a week. But we knew this thing was kind of coming, and we knew that this was, and we probably would have come back if we could have another time. But we knew that we had to collect all the data, so we were furiously kind of growing yeasts and taking photos and photogrammetry scanning them, and then that was it. That was all the data we could get. We could still have conversations, obviously, but that was the end of the experience in the lab. So it was a real rush to get as much raw material to work with as possible.
I was able to do like a crystal modification of the yeast and do that hands-on, wasn't I, working with Oscar? And I always say that the reason I want to do the hands-on stuff is because people want to ask me about it. So if I'm, for instance, over the past few weeks, we've been doing endless guided tours of the BioArt Revolution show in Timisoara, which we've just completed recently, which Sonja was also involved with. Everybody, really, nearly, and I'm giving talks to school groups and all sorts of people, biology teachers. You don't know who's going to turn up, the dean of life sciences of the local university or whatever, and they want to know what it is all about and everything. And if I haven't done it myself, because I learn by doing, so I need to do it so I can tell people what it is and how I did it and why I did it and what's interesting about it, and do it from an aesthetic and scientific perspective, I guess, so I can fuse all those things in one work.
Fantastic bread that you sent to us that became part of the. It's not on show here, but there's another part of the Fermenting Futures project called Culture, which is buildings covered in breadcrumb that was you made.
Right. Yeah. So I think it's a lot about materials, about the process itself, as you say. So being really involved in generating the materials also is, I think, an essential part of of the artistic process, and the materials that are available or those that are not available or might be necessary for to realize an art project. So that is one point to the potential frictions you mentioned. I would not call it frictions. I mean, that's what is fruitful in a good collaboration, when you have this kind of when you touch difficult points, let's put it like this, and grow beyond your state of the art. So one thing was, for those who have seen it, on the Fermenting Future flask, there is a piece of PLA, of polylactic acid, that in the end we have made in the lab from the lactic acid that was produced, and where Anna was involved also in making it. So Anna said, I need this PLA, and I said, well, that's not our experience. We can make the monomer, lactic acid, and then this is polymer chemists who polymerize it. That's not our expertise. We don't have a protocol. Then I said, okay, I need it. Can you do it? And we started to ask around, reaching out to colleagues and to the chemists, to the expert, and kind of no one really gave us a decent protocol, which honestly annoyed me a bit. And this is a moment that's very stimulating. So I said, okay, we're gonna do it. And in the end, we found a protocol and we learned how to make it. So we can show now the PLA, or Anna can show it on her artwork. And that's a point, I would say it's one of the unplanned but important, let's say, learnings or gainings from the science viewpoint. So when the collaboration brings you to a point that is like getting out of the comfort zone or something into unknown fields, and where you are forced to explore that and gain something new, learn something, acquire new techniques, etc. And I think it's very much, or maybe it's, I think it's a lot material based, or at least in that kind of artwork where that you're doing. And the other, like the bread, I mean, that sounds simple, make some bread, but it was with a special engineered yeast, and due to this project we learned that we had to further engineer this yeast, so that took some extra weeks or 1 or 2 months to make that strain, and then bake a bread with a grain that was already around like 3,000–4,000 years ago, and is still fortunately commercially available. So in the end, I baked this bread in the kitchen, dried it, and shipped it over to UK as material for you to
tell them about what you had to do.
Yeah, that was just the beginning of the nightmare. Well, one turned out that it was really mouldy, and then
it's been like 3 weeks in the post.
Yeah, and then we
had the Brexit story.
Yeah.
And it was this beautiful but very, very sort of dense loaf, and then we had to chop it all up, and because it wanted breadcrumbs, so it sort of really needed to pulverize it, make it into breadcrumbs. It was really.
We knew how to. The mouldy one, because I worked with the Centre for Emergency Preparedness and Response in the UK previously, and they told me how to sterilise stuff. It was covered in mould, so you had to suck off the mould with a HEPA filter and then bleach it.
Yeah.
Didn't we?
So we bleached the bread, and then we pulverised it, and it didn't just break. And then dried it for hours and hours and hours, and it was just this weird bleachy, bready, mouldy smell.
And the coffee grinder.
And then put it through. Days of coffee grinding, and I've never used that coffee grinder again.
You can see this piece if you go to the Nobel Prize Museum now, because it's on show there after all this struggle. It's called Culture, so it's on show there until the 7th of January, I think.
It sounds like that collaboration went always also in your private life and starting bakery career. But what would you say? I mean, the roles shifted, as I understand, also in between the collaboration. What would you say for future projects also, what do you think is like essential or necessary to start like a successful collaboration? And what would be like the roles and how flexible would they need to be?
If I may start, I would say the most important is to start with the right concept and right plan. And that is certainly, from my viewpoint, to initiate or plan a true collaboration. Not something like where one of the partners is kind of just like a mediator for a certain goal. So I'm thinking of like, okay, let's do something with art for our scientific public outreach. That is not the ideal approach. Or I could think of an artist thinking of, okay, let's use a lab and show up there briefly and just like put a tag on our artwork that it's science-based. So the true open collaboration and allowing the unforeseen to happen, be open for the unexpected, and then kind of see what happens. Sounds easy, but yeah.
Yeah, it's definitely right that about the not so much overseeing the goal, because I really like to, like when I start a collaboration, I all the inspiration comes from going to the lab and what I do and understanding the research, because I'm about, rather than imposing my idea, I'm about like getting the idea out of what I'm seeing. So it's like I guess a painter painting a picture of something that inspires them. I'm inspired by what's happening before me and the idea of telling that story or pushing that story. So it's that openness, not being controlled too much. And also I don't really like it when I have to write a proposal that says what I'm going to do. I usually say I'm going to come in and see what will happen. And I think that's the way you get the more genuine stuff coming out.
And just being enthusiastic about the subject. It's you know, the science, everything around the science is just fascinating, and just I'm just personally interested in these things and the subjects, and then the discussions that we have, and the way that we can sort of tie it into bigger pictures. I mean, it's just something that I'm really enthusiastic about, and I think unless you really are into it, you're not gonna fully sort of engage with it. So again, it sounds very simple, but it's if you choose your science that you're into.
But
what?
You can get into things that sound really weird and that you don't think you're going to be interested in at all.
Yeah.
Like I never thought I'd make any art about fecal bacteria, but I've made loads now.
Did you not?
No, I didn't.
I had a
No, don't tell that story.
But no, it's like your areas of interest and experience and knowledge, are shared across disciplines, and I think that's the sort of stuff we're talking about now. Is that there is a shared ways of thinking and tools between art and science. They're not that different, really, in terms of what their goals and aims are. So I think if you're sort of venturing into those areas, then you can sort of just keep spreading out and keep feeding into sort of stuff that you've done.
Yes, I'm quite flexible with the work I'm doing, but it came from my career. Like I worked as mediator, facilitator. Anna forced me to be a curator. I succeeded with you again with BioArt Revolution. It was really a pleasure. Thank you. But for collaborations in general, it's really like Diethard says, if everyone is joining this team, you will find your position where you give something fruitful to it. And the transformation, the the negotiations, the process, this is what we love to do. And sometimes we don't know where it will come to. It never has really an end, to be honest. So even if there is an artwork, it doesn't mean that it's finished, and this is what I like most.
Yeah, I think the way the project was built was an adventure, but it was an adventure because there also was COVID. But I would say, if you look on a more like general perspective, how should art and science collaborations or residencies look like? Then I would say it would be good if there would be funding bodies giving as serious money to those projects than I would say to scientific research, because everything we did was like next step. Of course, there was a plan, there was a concept, but sometimes we didn't know, do we get the money and how much and how long can Günther Seyfried work in the lab at Boku, for example. 12 months was a long time, much longer than Anna and Alex did, and but in the end we had no time to publish something. So so this, I think the outcome, if you look or say the successful collaboration, if we may say, you can see here, but it was difficult because of the pandemic and because there was funding, but it would have been much nicer if there would have been, I would say, a deeper understanding of the process in the first place, and having funding bodies that would step in and say, super, this is what we were looking for, but it wasn't. So there was a lot of work by each individual contributing to make this whole complex project possible.
Yeah. So I think like the success also of this project was your intrinsic interest and energy engagement you put into it. So thanks for that, and also looking forward for the next projects. But maybe before we dive deeper into like the project itself, I would like to also open up the panel for discussions from the audience. So perhaps anyone would also like to know a bit more about specific collaboration or also the roles, because we have also a lot of people here in different roles working on art and science collaborations.
Hello, thank you very much for that, guys. It was really fascinating to hear, especially the different stages of going through the project. So I think that's really informative for a lot of us in here today. Kind of two small questions. The first one is more for the art science kind of actual collaboration. Could you elaborate a bit more on to what extent then did you as a scientist become an artist in that process? And were there ever any moments where you curated the art, or was it a constant collaboration all the way throughout? Were there moments where there were more clear defined roles, or did the scientist then become the artist in some aspects? And then the more logistical stuff, when it came to actually organising the, you mentioned earlier the difficulties of getting into art galleries and scientific spaces, and specifically art galleries. Was there any moment where perhaps you thought that you tried to get it into non-traditional spaces? Because thinking about the larger conference, must we always rely on artistic or scientific spaces, the spaces where we actually display and project this work? Or was there any moment where you thought of, of non-traditional spaces other than artistic or scientific ones to either look for funding or to display the works?
First of all, it was a wish from the group to show artworks in art museums. I thought it's super easy. I know in Vienna so many curators and museum directors. I had really many coffee talks, super interesting work. Wow, the Boko is inviting artists. Then it was like, maybe you go to a small project space, and we said like, we don't want to go to a project space, we want to make an art exhibition. I know project spaces, and I know it's very good and easy to exhibit at off spaces. Yeah. I think in the future, in general, the museums can develop, yeah, because it's not just that you say like, oh, art-science collaborations should be just in technical museums, natural history, of spaces, because there are really narratives which bridge to contemporary art, to ethical, philosophical questions, and why is an artist suddenly forced to make an artifact for a certain room? And we cannot change the room, but we can provide museums with knowledge, what artists need who are doing new media, and they need to improve. So maybe they invite more of the curators here to give an expertise, because they're working like 20 years in that field. So we have a knowledge. But yeah, we need to work more on the topic.
Okay, to your first question, I'd say as humans, we like to put things and people and everything somehow into boxes. So there is a box for scientists and a box for the artists. And there is certainly, in my opinion, let's say a certain grade to which a scientist can be also an artist, or I mean scientists, there are scientists that are artists in full sense. And one thing is that, of course, it changes the interest for scientists also like to actively engage in art. And on the other hand, Anna is an example of an artist who has a formal education also to work in the lab. And
informal education.
Or informal, but I mean formal in the sense the basics of the safety instructions, etc. So yeah, to a grade, I would say you are actively engaging in science. I would say I would not make the claim, and probably it's much easier as a scientist to make the claim, okay, well, now I'm an artist also, than an artist would say, okay, now I'm a scientist as well. But it brings knowledge and know-how together, and at a gradual level, certainly these collaborations change the abilities, change the mindsets, and that's what is very important also directly for the people who engage in such collaborations. I don't know if that's a valid answer for your question or any helpful in any sense. Maybe Anna, you can add on that.
I mean, I was come from the art perspective, but I see art as a kind of meta-discipline that can bring together things like history, philosophy, ethics, science, everything, and try and tell a sort of story that reaches people on an emotional as well as intellectual level. So that's kind of what I'm going for. I wouldn't claim to be a scientist. I can perform sort of scientific acts. I can pipette like the best of them, I guess, relatively. But I can certainly not infect myself with pathogenic bacteria, because I'm very good at that. I've done that. I've avoided doing that over the last 20 years, so that's good. But I mean, I think, Yeah, art as a kind of meta discipline. There's this idea of transdisciplinarity, and that's where you blur the methods. So art done through a scientific methodology, science done through an artistic methodology. And I'm very interested in that. So that's always kind of happening when we're doing this stuff, isn't it? So then it gets kind of blurry. I don't know if Alex has any comments on that. Something deep.
Yeah, something deep. I mean, from my perspective, because Anna does the hands-on stuff. I would never, I sort of stand back and just observe.
You do hands-on if it's by computation or something.
I do I basically do the digital stuff. So and because science has adopted digital tools. I have been able to have conversations with scientists and in labs that I wouldn't have been able to have 20 years ago or more. So it's a sort of shared knowledge of techniques, of algorithms, of approaches, of having done a lot of computer vision stuff and big data processing and for art. But they're using it for science. So this is why I sort of tend to come at it from we're kind of we're using the same tools in roughly the same direction. For me, it feels there's a real parity through a digital lens that there's more similarities than there are differences. Maybe it's a very sort of digitally orientated viewpoint, but that's my background and interest in it. So for me, I think there's, if you have a grounding in computation, if you have a grounding in these digital skills, there's a real commonality, and that acts as a sort of way of smoothing over the lines of differentiation, which I've come to just see as kind of folly and a very outmoded way of thinking about people and what they do and things, and yet we're still doing it in schools, and we're still teaching subjects and telling people they can't do things later that they might want to do because they're one or the other. And I just think it's a very something that needs changing, but it's a very deeply ingrained situation. So maybe this can help, and we can all spread that kind of idea.
Is there another question? Yeah, I think just behind you, you can head off the mic. Thanks.
I suppose my question is about critique and how critical an artist is of the technologies they're embedded in. And I work closely with, I'm a social scientist, I'm not an artist, but I work closely with scientists, so I ask myself this question the whole time. But Anna, you said that we like this interesting twist where you could end up being drowned in plastic that was actually produced from carbon dioxide from the yeast. So I see that as being a kind of critical, sort of playful angle on the science. But how critical do you want this work to be of these technological solutions? Because biotechnology is not the only way of solving the climate crisis.
I mean, the problem is that people are very dismissive of the biotechnological approach as a solution. And actually, I think we need a bit more balance on that, it's not, in some places, it's not allowed to sort of suggest that, well, maybe genetic modification of food or something like that, because we did this other project, Biotechnology from the Blue Flower, which was about genetic modification of plants to produce inulin. And it's this idea of like, we're at this biotechnological revolution, and maybe we need to have a bit more balanced debate about this, because for instance, in the UK, it's like you're not even supposed to say it. We've done projects with certain large organisations who are very much to do with ecology, and I know the people that work in these institutions want to talk about biotechnology, they want to talk about CRISPR, they want to talk about DNA modification, and the powers that be that are in charge of them aren't even allowing them to speak about it. So if we can't even speak about it, how are we going to consider these or have an ethical debate about whether we want these kind of things? So, I mean, the
the idea behind the work is that you could drown in plastic and you would solve it with this kind of loop of the you know, the carbon being reabsorbed and then made into more plastic. So it's like throwing that idea out there. So I don't tend to take on projects where I'm actually critical of the science. Like, I don't think it should be done. I do think it should be done. I'm interested in allowing the public a way into discussing it. I think that's what I want. I want them to actually be able to understand and participate in the debates rather than just being shut down. Oh, you can't do this. This is the way to solve it. We've just got to sort our rubbish into different piles, which doesn't work anyway. If you look at the kind of the, you know, the stuff behind that, it's one of those things where we're kind of told that if we just did these things, then everything would be fine, and it's not the case. So I'm interested in this, like, broadening of a critical debate there, I guess, if that's not too controversial.
Yeah, I think that's exactly the point also for science, or what science should aim for, and in most cases I would say does, and often does not really get that through. This open discourse about, let's say, where certain research can lead us, and to this specific topic, like climate crisis, there is nothing like one solution, and we will need a lot of different solutions. And as scientists, we can just like try to explore if a certain route can be one piece of the solution. And if we don't do it, we will never know. So that's the first thing. So the first important aspect in that type of research is to explore where a certain line of research and development may lead us, and an open discourse about the possible consequences is extremely important, of course, and that's exactly what Anna meant. And often, yeah, this doesn't even happen, as you say, also because certain things are kind of out of the debate quite often, like the GMO topic. And debating it does not mean that this is what should happen, but it's one possible scenario, one possible future. Like, more discourse about these possible futures, or let's say possible technologies, would be really important. And if art can be another medium to enable more of this, we have gained a lot as a society.
There's another question in the front row.
Yeah, thanks a lot for your fantastic collaboration, because you seem to be very harmonic, and it's not always the case, and it's very good to see a perfect match on stage. Just before visiting your exhibition space, and it's very well designed, and it's interesting because just in front of your exhibition space you have this infinity engine. I don't know if the curators of ZKM did this on purpose, but in the very front of the piece there is a video running by Oren Katz, who in the video, by the way, gets a doctor. He's not a doctor, and he was a professor, but apparently if you are having a scientific discourse, you add it a doctor quite easily to give you more importance. So that's what institution often did. I was reminded of a question that we got, Oren Katz and myself, in 2003, really 20 years ago, when we were working on the exhibition Art Biotech in Nantes in France, where we put together the forerunners of biotechnological art forms such as George Gessert, Joe Davis, Eduardo Katz, and also the Tisha Kalshan Art Project, who did come up at this point with a project called Victimless Leather, and victimless food and disembodied cuisine as growing frog steaks for consumption and to spare animals from slaughter. And we got a question from the audience, and that's a very British question I want to ask you. Because we had a question from the audience from the last row. We didn't know who these guys were. And then it turned out that there was a question, that is not art, that's just design, you know? And the person asking this question was Anthony Dunn. And he was at this time in the process of creating speculative fiction and speculative design, while nobody of this team at the time had even touched a Petri dish in 2003. And then they went on to the wet labs and tried to combine the speculative design aspect with wet hands-on practice. So it was interesting that it was coming up, and Oren Katz, who is speaking as a doctor now in this video, replied, no, I spent my whole life to get away from the application-based positivistic spirit of the design world, application-based demonstrations of applicable solutions that are aestheticized through the arts. So I don't want to build up another dichotomy again, but of course art, design, science, engineering become so much merging. So, and the question was brought up by the previous question about the criticality. So the criticality of the work, you know, and in Symbiota's work at the time, it was because there was this hyperbolic claim that you can grow victimless meat, and you, of course, you cannot, you know. And there was all this discussion about the fetal calf serum that you need in order to activate the cell growth. So the medium was a message. So the growth medium became the message, and this was the critical aspect to it. So I just want to ask this question 20 years later, because we are now accustomed to all the hands-on practices. So is it art or is it design?
It's definitely art, because I actually get quite frustrated with design. I don't want to give my opinion on design, but it's not something that interests me. I trained in fine art, so I come from a sculpture and fine art background. So that's always the kind of direction that I'm going to. But I think in terms of the victimlessness, it's quite interesting in terms of the yeast biotechnology, because you're actually achieving things that are carbon neutral from recycled products and things, and that's something that's very key to it, isn't it? With the, like, for instance, with the succinic acid.
Yeah, in that respect, I mean, that's it. It's maybe a side aspect, but what you mean is essentially what we might, or our processes might produce, is essentially made from a carbon source, and that could be CO2. From an energy source, and that could be electricity, and from some mineral salts. So essentially, really like a new way of production. But then, of course, we have to consider what we are producing and why. So a dangerous route would be like, finding an easy way out of the raw material crisis, just to go on as we do by now, or thinking of maybe not plastics, but fuels, if we can produce aviation fuels in the same way or in a similar way, bio-based, okay, is it reasonable to fly around like crazy as we are still doing today? Or that's the kind of discourse we have to take, and that's not a reply to your question directly, but in a sense, maybe also to a question that was raised in the beginning. Are the scientists, or have we as the scientists curated the artwork? So I may phrase it a bit different. If it would be design, we would legitimately say, okay, we want something, this and that, make us proposals, and we take this design that we like. We started, I think that I said that explicitly, you are free to make what your artwork is. So we are not interfering in, we are helping, we are supporting, we will tell you what is possible or not, but we are not telling you what to do or what not to do. And I think that is the main point, and that's probably part of the answer also to your question, is this art or is it design? And I would say clearly it's art, and that was the intention from the beginning.
I'm curious about the support in the science community. So how common is it that your colleagues will say, they may go beyond saying, well, this is nice, it looks, I'm kind of intrigued, who say, well, I give up some grant money for this, I think this is absolutely necessary. versus how common is it that scientists might say, this is frivolous, this is a waste of money?
Well, I guess there is a group of people who would consider it waste of money, but I'm seeing also a change. So in discussions with people and in a process of like also discussing potential new projects. What I see is that once you expose people to the process and to the outcome, slowly there is a change in mind to the aspect that, at least maybe a person is not really appreciating it by themselves, or it's not touching them, but at least understanding that this is not wasting money. If that is
good
an
to
answer.
hear.
I mean, we Sonja and Alex and I had an experience recently in Timisoara, because we were taken to meet the one of the head of life sciences university, and at first he was very resistant to the idea of art-science collaboration. Like, look at her face. So that's how resistant he was. And then he came to the opening of the exhibition and invited us all back to the lab and said, well, I realised I was very sceptical before, but this is the real deal. And we're going to have a conversation with all our teams, and we're going to see who wants to collaborate with an artist. And I think we know where we can get some funding for it. So he went from, like, this is a load of rubbish, basically, you're all mad, and why have you brought this artist who's talking about extracting plague DNA into my office, and to actually wanting it and discussing the possibility of studying zoonotic diseases in wolves in Romania, which is something I quite fancy doing. So you know, it's like, I think it's like having people see it happening and seeing what we really do. The more they can get exposed to it, the better.
And that sort of ties into the question earlier about why show it in museums, why show it in ZKM, why show it, because it brings it to audiences, it brings it to, you know, it sort of contextualizes it within those institutions, within those spaces. And that means that other science institutions can see that happening and will encourage them to, you know, see that there's a high level of outcome. So, yeah.
I think there was one more question. Yeah.
Yeah, thank you very much. I would like to pick up on that point, and I was wondering during your discussion, exchange, about how to leverage these experiences and how to make them not only more visible through exhibits, but you said you couldn't really publish. So I take a point that I heard from Ingeborg earlier today about education, and you also mentioned that you had a kind of presentations or lectures about this. Is this a way to leverage these experiences? How we make them more accessible? How we can have more of these projects in the future? Thank you.
Maybe I can say something. It wasn't just Diethard, there were other professors from BOKU involved, contributing to one of my books or inviting some of my students like Klaus Schöning and others. So I think this was a very good first step by Diethard, because Diethard has, I would say, a strong standing in the field. So he did it, you know, like as a very famous professor at BOKU. I wouldn't say the first step, but there were other professors at BOKU then following, and they, in a way, they were just waiting to a door to open, and then and then I think it became a kind of community. And whenever some students want to go to a certain lab with a certain facility, they call the people at BOKU, and this is spreading. And there are other facilities in Vienna where students can go for 10 euros a day, where they can do CRISPR and all kind, the Vienna Open Lab. And so I think it was, you know, like a first good step, and then other academics followed. And I would like to say one more sentence to the question, why would science not, like predict the future or visualize the future as much as probably art. And I had a couple of conferences in Berlin 10, 15 years ago about the future of evolution. It was before CRISPR became known. And most of, I would say, of the scientists, really peers in the field, said, well, science is unpredictable. And most of them were not willing and able to articulate their vision of the next 5 or 10 years. And in the conferences, I also had, of course, artists involved, and they were the only ones who were, I would say, eager enough or had a kind of qualification from the, art world to design, to imagine, to articulate something we can address as a futuristic vision. Could be positive, could be negative, and I think we hardly see this in the science world. And I don't have an answer to that, maybe Diethard, but I think therefore having the artist also involved, because at the art schools this is more common than in the science world, to speculate and to speculate about the future scenario, positive or negative.
Yeah, that's a very good point. In Vienna, we say if you have a vision, you need a doctor. But I would say science without a vision is lost. And I thank you really for that comment. So I think that's extremely important. Also, that scientists are encouraged to let's say, project visions or scenarios, and we must be aware, of course, that's a thing in the scientific public discourse often, that scientists are expected to have a very clear and distinct single answer to future aspects, and that's not possible, of course, because every prediction into the future is getting fuzzy or has certain or uncertainties. It's like weather forecast, it's the same thing. The further in the future, the more uncertain the answers are, and there may even be different scenarios, but without these scenarios and like navigating through them, we would be lost in the space of work we are doing. You can do, of course, fundamental research on this and that without a vision, but my personal opinion is that that should not be the approach, and it's possibly a great opportunity also out of these collaborations that scientists are getting support and encouragement to speculate or to project scenarios.
And I think, if I may just say one sentence, and this is more or less a quote by John Schellenhuber, which is a very important figure in the climate change debate. And he started having in Potsdam 20 years ago a lot of artists, some bio-artists, but not many, but artists from the performing arts and so on. And the question was, why having people from the creative sector and artists getting involved in the science, in the science world? And the argument he had like 20 years ago, I don't know what he would now say about that, but the fear was if scientists would speak up and come up with a vision or with a critical perspective, or saying we are the ones that are responsible for this and this and this, then the politician wouldn't take them as serious as they were taken by not speaking up in that direction. And so they invited the artists and curators and so on to give them, or to give their research and their fear about the future and their criticism a voice. This is not like science goes public through art. This was a different perspective, but it was all about the integrity of the scientists or the science community. 20 years later, I think the situation is a different one, but there were good reasons, I guess, by Schellenhuber to have such, an argument 20 years ago when he had and Schering Stiftung was also involved. I think it was called Tipping Point. It was a huge project. And yeah, this just came to my mind when you just elaborated on your topic.
Yeah, I would say thank you so much for giving all the insights and inspiring starting points also for new discussions. Maybe before we wrap up the round, I'm ending with a quick question, because it worked quite well with you in a collaboration. If you could work in the future on another topic, what would it be? Like one word, maybe.
You mean in art science collaboration?
Yes,
or in
yeah, in this, maybe also in this constellation. Yeah.
If in the same constellation, okay, I was tempted to say I would explore a very different art discipline, but then probably it would, maybe with Alex on something real digital art, let's say, non-material.
I'd like to work on pathogenic yeast, the really dangerous ones.
Yeah, there can be mean ones. Yeah.
I'll work with Diethard on that. That sounds fun.
I can add that we will have another residency with Anna next year, so you will get an answer. And Alex, sorry, yeah, Anna and Alex. Next year you will get the answer.
Well, if we would go with this, you know, like bloke again, with this mishpoke, I would say, no, the only thing I would do differently is like documenting more, because there was a lot of, I wouldn't say fun, but the thing is, like, we learned a lot. We learned a lot on many, many different levels. And the only thing what we have now is a kind of object or an outcome, and the whole process behind that, and all the lessons we learned from the science side, from the side of the history of evolution of mankind and all that. So because we had a huge field that we walked through, and this is a pity that in the end you have only a kind of object, or
only the object.
Yes. So and yeah.
Wow. We did write some papers, though, didn't we? We did. We did write some pages.
Well, let's see. Maybe it goes on with the yeast topic. At least you're still intrigued with it. And so, thank you so much for tonight and for your interest. And I hope to see you soon. So, good night.